I'm going to be reviewing the post-transcriptional processing which occurs inside the nucleus. It is important to note that mRNA polymerase lacks the ability to proofread its transcribed mRNA strand. Therefore, it is unable to detect and correct errors. This is not detrimental since the errors are not passed down to offspring. In addition, most genes are transcribed many times, thus reducing the severity of one error. An mRNA that has not undergone post-transcriptional processing is known as a primary transcript, pre-mRNA, or heterogeneous nuclear RNA, also known as hnRNA. During transcription, the 5' end of the mRNA is capped using GTP. This cap is known as the 5' cap. This serves for two purposes. One, for the protection against exonucleases, which are enzymes that cleave nucleotides, and two, to serve as a point of attachment on ribosomes for translation. A poly A tail, which consists of adenine nucleotides, is added to the 3' end of the mRNA. This is also referred to as being polyadenylated. It helps transport the mature mRNA to the cytoplasm after splicing and helps initiate translation. Splicing removes introns from the mRNA strand. They do not code for proteins and end up being discarded inside the nucleus. The mechanism involves using several small nuclear ribonucleoproteins, also known as SNRPs, which contain proteins and small nuclear RNA, also known as snRNA. Therefore, snRNA acts as a ribosome, which is an RNA that is able to catalyze chemical reactions. Splicing occurs when SNRPs recognize the nucleotide sequences at the ends of the introns. They then pull them together, forming a loop, also known as a lariat, and are then spliced by the spliceosomes. The exons are then formed together by the spliceosomes, thus creating the mature mRNA. Alternative splicing is a mechanism that is used to help regulate genes. It creates a variety of mRNA molecules for translation using one DNA coding sequence. Therefore, it contributes to the diversity of protein products. By leaving out certain exons, incorporating certain introns, and using different splicing sites during the post-transcriptional process.